Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Navigating Complexity, Tailoring Cell-Based Bioassays for Advanced Therapeutics. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Solvius, who developed the content for this presentation. Solvius help innovative companies advance their path to commercialization with CMC analytical solutions powered by deep scientific expertise and a relentless focus on customers' success. With years of expertise in small molecules, biologics, and cell and gene therapies, their talented subject matter experts have the know-how, creativity, and tenacity to solve the most complex analytical challenges. Now I'd like to introduce our speakers for today's event. Zhenghua Ye earned her PhD in immunology at Aix-Marseille University and received her postdoctoral training at Stanford University. At Solvius, she leads the bioanalytical strategy for drug release testing, ATMPs, and large molecules, and ensures regulatory compliance for the cell-based bioassay department. And Dr. Reg Clayton is the head of biosafety at Solvius and deputy site head of Solvius based in Homburg, France. Reg has worked in various positions in the pharmaceutical sector for around 25 years, including as a project leader for small molecule antivirals, discovery at Johnson & Johnson, principal scientist for virology at Merck, and vice president of serology and disease screening at Quotient. But without further ado, I'd like to hand the presentation over to our first speaker for today, Zhenghua Ye. You may begin when you're ready.
Okay, thank you, Ryan, for that nice introduction. And hello, everyone. Thank you so much for joining us. Over the next 40 minutes, we will dive into three key aspects of potency assays, from drug development through to commercial release testing. First, we will cover the regulatory perspectives. Then we will focus on cell-based potency assays. And finally, Reg will walk you through the potency assurance strategy. And one quick note, due to a lack of a client disclosure, we won't be able to discuss the case study on potency assay of ADC today. And we apologize for this inconvenience. And for those of you just joining, welcome again. Now, if everyone is ready, let's get started. Just a quick introduction of Solvius. As a spin-off from Novartis, Solvius was founded in 1999 and has broad expertise and experience across pharmaceutical analytical development and testing in small molecules, large molecules, including ADCs, and also advanced therapeutics such as CGTs, cell and gene therapeutics. Our group comprises two sites in Switzerland in Geiselhorst and Basel. Homburg in France, where most cell-based potency assays are performed. One site in the United States in Canton, and one site in the Netherlands. And due to the high demand of our service, a brand new facility was acquired in North Carolina in the United States, and where the laboratories will focus on the analytical testing of large molecules and cell and gene therapeutics. At Sovias, we work with more than 700 companies per year, from CMC to commercial release drug products. And more than 20% of our colleagues have a PhD degree and with excellent tracking records in publications and international patents. So today, the outline just showing you here that we will first discuss the regulatory framework for potency testing in advanced therapeutics. And then I will discuss the selection of cell-based potency assays. And finally, Reg will walk you through the potency assurance strategy. So first at all that we will discuss regulatory framework for potency tests in advanced therapeutics. So what is the potency testing? So potency testing is essential in drug development and the CMC process. So this potency testing are the measurements specifically designed for testing the ability or capacity of a particular drug product. And how to design this potency testing? So first thing, we need to understand the basis of the diseases, where the drug is going to apply to, and also the target patient population as well. So all this will correspond to the TPP, the target product profile, where all the product characterization will be summarized in this profile. And also the potencies will be able to measure all this potency according to the expected mechanisms of actions, these MOAs of the drugs. So potency assays are a critical component of drug development, particularly from a regulatory perspective. These assays measure the biological activity of a drug product, providing essential information about its efficacy and ensuring consistency across different batches. And regulatory agencies such as the FDA, EMA, and ICH require comprehensive potency testing to verify that the drug performs as intended as its therapeutic dose. So accurate potency assays are vital in demonstrating that the drug's quality, safety, and efficacy remain within the required specifications throughout its shelf life. So in this slide on the left side, as you can see, when the drugs go into the clinical trials, that's we so-called the investigational products. So throughout this process of the development of this drug, so the drug data will have to be able to show that the identity, the quality, purity, strength, and also the stability of this drug product. By establishing a direct link between the potency and as well as the clinical outcomes throughout the clinical trials, these assays help ensure that a product meets the necessary regulatory standards, facilitating the market approval, and most importantly, that maintaining the patient safety. So as a drug product progresses toward a commercial release, the criteria for potency assays become increasingly stringent. This is necessary to ensure that the product consistently meets the required quality, safety, and efficacy standards. The following key criteria are typically emphasized and tightened for release testing of the licensed biological drugs. So as you can see on the right side of the slide, those are the requirement criteria for release testing of a licensed biological drugs. First of all, this potency assays will have to be able to demonstrate the potency that is specific to the drug product. And then these potency assays will provide the possibility to assess the so-called critical quality attributes for the drug release. And also this data, in general, they are quantitative data. And this data will have to be able to meet predefined acceptance and or rejection criteria. So this also included this most importantly is that appropriate reference materials, standards, and also the controls in these potency assays. So all this data that will be able to establish and document the accuracy, sensitivity, specificity, and reproducibility of all these test methods throughout the formal validation process. And this will measure the identity and the strength, the activity of all active ingredients as well in this potency analysis. So this data will provide to establish a dating periods, and finally will have to meet the labeling requirements for the drug release, especially for the licensed drug products. So in following this guideline is that what kind of the analytical methods that can be used in the potency testing development. There are two major assays that can be applied to the potency testing. The first one is the biological assays, and the second one is the non-biological analytical assays. So for the biological assays, that's also the assay that we called the bioassays. So these bioassays are quantitative, and that can help us to evaluate a product's active ingredient within a living biological system. So a living biological system, which means that there are two types of the system that can be used. The first one is the in vivo animal models. And then the second one is the in vitro, the organ, tissue, or cell culture systems that could be in vitro or ex vivo as well. But in respecting the 3R rules that tell us that if we can find any other alternative methods that to limit the animal use where possible, which means that we will, if we can find the assay that can replace and reduce and also refine the animal uses in research, teaching, testing, and exhibition, and that's better for these bioassays. So this is why in today's webinar, we will mainly focus on the in vitro the in vitro organ and tissue, the cell culture system right here. So for the non-biological analytical assays, so these assays are considered as surrogate measurements of the biological activities, such as the enzyme activity, and is a reductionist model as compared to the biological assays. And in general, that these non-biological analytical assays will be able to provide us the extensive product characterization data that could be including the immunochemical attributes, biochemical attributes, molecular and genetic attributes of these drug products. And this assay data will be correlated to a relevant product-specific biological activity, of course. So this is why it's more like a surrogate model as compared to the bioassays. And in case of that, the product has complex and or not fully characterized mechanisms of actions, or the product has multiple active ingredients, or multiple biological activities or with the limited product space stabilities or biological assays is not quantitative and not sufficiently robust or lacks of a precision. In all these cases, probably we will have to consider to develop an assay that's called the assay matrix, which means that we'll have a couple different assays together. So these multiple complementary assays will have to show that the relative potency of this drug to measure the product attributes that are associated with the drug quality, consistency, as well as the stability. So as I just mentioned that for all these bioassays, they are more relevant to the in reality in the living system. And what are the benefits of using these cell-based potency assays? So there are three main benefits of this cell-based potency assay. The first one is that most of them are comprehensive. They are approximate the complexity of the biological systems. And in some of the potency assay, cell-based potency assays, they are relevant to a wider range of drug modalities, such as large molecules, small molecules, as well as the cell and gene therapeutics. And also, these cell-based assays, they will enable us to have a look into the functional analysis and to provide the insights into the dynamic bioprocesses. For instance, like the cell uptake of the drugs, and also once the drug or gene go into the cells, we look into the translation, for instance. And also, this potency assay, they represent the behavior and also the reactions of the whole living organisms up to months. Some of the cell culture systems that can be set up and grow for a couple months, and some of them are shorter timeline, but that can be that could be longer than weeks and months. So they are quite relatively comprehensive as compared to the biochemical assays. And the second, the key benefits of the cell-based potency assays is that most of them are versatile, which means that if we set up a cell-based potency assay as earlier as we can during the drug development, then we can probably detect the off-target effect as early as possible, or any of the toxicity of this drug as early as possible in these cellular systems, such as some of the small molecules that we can determine the maximum tolerated dose with the cell cultures. Or with some diseased modeled system, we can also look into the minimum effective dose as well. So they can be very versatile in these potency assays with the cell cultures. And the traditional two-dimensional, these 2D monocultures methods, they are a long history of mimicking the in vivo cell growth. However, this method cannot fully represent the physiological conditions, such as that they are incapable of replicating the essential cellular communications between cells to cells and the cells to the extracellular matrix, and also the cellular responses to dynamic mechanical stimulation, such as blood flow. So therefore, to solve these problems and challenges, in the past few years, there's quite a few 3D cell models have been gradually developed to provide a 3D matrix-like structure for the analysis of the cell attachment, proliferation, differentiation, and communication in static and dynamic cultural conditions. And also, most of these cell-based potency assays, they can be easily scaled up for multiplexes and also into a high throughput format as well. So with the high throughput format, we can collect more data, and that can really help us statistically to determine the EC50 and IC50 of the drugs in this assay system. And then the last benefits that we can think about is of the cell potency assays is the speed. So these cell-based potency assays, they are relatively straightforward as compared to the in vivo animal models. Some of them take a couple of days, some of them take a couple of weeks. So they are relatively straightforward as compared to the animal systems. They could be diverse, but they are rapid and robust endpoint readouts with these potency assays. For instance, that the most measurements, the readout that we measure regarding the endpoints could be the absorbance or fluorescence or luminescence. So they kind of a relatively easy readout as well. So in the next couple minutes, I will discuss about the selection of cell-based potency assays. So the diversity of a cell potency assay as showing you here, those are the cell-based potency assays that we have been working a lot in our routine assay development and also in the drug batch releases as well. So for instance, we determine the cytotoxicity of the drug products. And to measure the cytotoxicity, we can perform the Nexin 5 or 7 AAD. That's a very simple flow cytometry analysis of these cells. And we can also look into more specific signaling pathways, such as a apoptotic pathway. So in that case, we look into the activation of the Caspase-3 and 7 pathway, for instance. And in this cell-based potency assays, very often we look into the cell viability. So for this step, we look into the metabolic activity, such as the mitochondrial reductase activities, and also the cellular ATP productions to look into the cell viability. And most of the case, we look into the cell proliferation as well by measuring the DNA replications, or probably could be the GFP labeled the cells. We counted the GFP positive cell during the proliferation process. And sometimes we can even look into the differentiation of the cells to look into the lineage-specific markers expressed on the cell surface. So in this cell potency assays, we can look into the cell signaling. Sometimes we look into the secondary messengers, such as the cyclic AMP. Cyclic AMP plays a key role in various cellular signaling pathways downstream of G protein coupled receptors to the intracellular targets. And this cyclic AMP influence a wide range of physiological responses of the cells, though that has been often measured during the potency assay setup. And for the intracellular calcium fluxes, for instance, to look into the T cell activations, that's often being used as a readout. And for the signaling pathway, we can also look into the receptor transcriptional activation as well downstream of the stimulations. For some of the drug modality, we can look into the blocking. Then we will look into the reporter gene inhibition in this case. And the other pretty much often used to look into the cell migration in the potency assays is called the scratch wound assays. So those assays has been used a lot just to look into the cell migration, cell proliferation, as well as the wound healing process after the drug treatment. And for the most of the lymphocytes, for the immunology part, we have been working with a lot is a so-called transwell assays to looking into the chemoattractants of the cells migration from the top wells to the bottom wells. And some of the potency assays, we can look into the vaccine delivery. For instance, that we co-culture the cells with the liposomes. We look into how the molecules or RNA or DNA will deliver into the cells. So those has been used a lot to look into the efficacy of the vaccine delivery as well. So finally, that is also the most often used the potency assays just to looking into the cell binding. In general, is the peptide or antibodies binding onto the cell surface. Or if that's a competition assays, that can be also to determine in its potency assays to determine the target engagement just right here. So this is the most used the cell-based potency assays that we have been in-house here at Sovias to look into for the batch releases. So now, how can we start to develop these cell-based potency assays? So most of the cell potency assays, we will require the cell line as a reporter system, for instance. So in this case, that we will start from selecting the best suitable cell lines. So these cell lines will express the specific target. And then most importantly is that not only the target expressed on the surface, it's also the quantity of the target expression on the surface is important. That will have to be relatively correspond to the inherent expression level on the targeted cell populations in the patients. And this cell line, we will select them, design the method that will correspond to the drug mechanisms of actions. And the stability of the cell line is important. If we can avoid to culture the cells with the antibiotics, that will be better. Since any of the antibiotics added to the cell cultures will increase the stress of the cells. So this will introduce the variability in the assay development. So this is why that if we can avoid to have the antibiotics in our cell culture, that will be a better choices. And the last point, and also is the most important point, is that we will have to be able to ensure that the cell line we selected, they have a legal aspects that will allow us in using this cell line in our assay development and for the drug release testing as well. So once we selected the suitable cell line that we can use for our assay development, then the first thing is important is to generate the cell banks. So first of all is that we will start to generate this master cell banks with the lower passages numbers. Of course, once we have the cells, we will test the mycoplasma and the sterility of the cells in the beginning. And during the cell bank generation, before or the end, we will also test the genetic characterization if we could. And if the project is needed that the genomic DNA of the cell line has been modified, then that will be better to ensure that the genetic characterization will be done to make sure the genetic characterization showing the stability of the cell line as well. So all this characterization, genetic or the phenotyping characterization will help us to construct or build up the cell line authentication. And once we have the master cell bank prepared, ready, and we can start to prepare the working cell banks. Once again, that the mycoplasma and sterility will be also tested with the cell banks to at least the end of the productions. That will be tested as well. So the format of these assays that could be a continuous cell cultures, which means that the cells, we will passage the cells once or twice per week. And the materials is in a consistent quality. We will measure to ensure that the phenotype of the cells is not changing during the cell culture processes. So all this, about 5% of the cell banks prepared will be tested. And ideally lower passage as possible, just to make sure that the cell lines maintained the same phenotype throughout the time. And we will QC the cell line as well. So the other format for the assay development is called ready to use. So this format is preferable. Why is that? Because they are ready to use. We just throw out the cells, we incubate with the drugs, and ready to go for the assays. So, which means that using the ready-to-use working cell banks will help us to increase the operational flexibility, which means that we will be more flexible based on the timing. And also that can improve the assay reliability as well, since we introduce less variability in our assays right here. So now we have the cell line selected, and we have a comfortable number of the cells in the cell banking. So we can start to design our potency assays and be ready for the validation of these potency assays. What are the considerations that we have to take into account is that we have a sufficient number of the replicates for the statistical analysis throughout the study. And also the sample setup in the assay will be randomized to reduce the biases that we introduce in the assays. And also, since all the assays are specific, we will have to identify and limit the sources of the variability as much as possible that can influence the performance of our assays. And also, we have appropriate assay-specific reference materials and control. Most of our potency assays are semi-quantitative, which means that we have to compare to our references or the reference standards and controls. So this is why to have appropriate assay-specific reference materials is extremely important to think about it ahead of time for the assay design. And we have to ensure that this long-term availability of all these critical reagents, which means that will include the cell banks, include all these culture medias and the growth factors, and all these reference materials and controls. They will have sufficient numbers for the long-term studies. And for the criteria of this design of the potency assays, that most of you already know that. The first after all is the accuracy of the method. The second one is the precision of the method, to look into the reproducibilities, repeatability, and also the intermediate positions as well. And also that we have looked into the specificity of the assay to be sure that everything we measure in these assays is specific to our drug products. And also we use this assay to determine the linearity and the range of the detection levels. We determine the limited of detection and limited of the quantifications level based on the working range of the method. And finally, that we will look into some small changes in our method to reinforce that robustness of our assays that can be performed as robust as possible. And then finally, once we all think about all these criteria and we have all the reagents ready, now we can think about the validation plan. So first at all is that for the validation, we will have a sufficient description of our method that we choose with a justification and also scientific rationale why we use these assays to answer the questions of the potency of our drug, and also apply sound and appropriate statistical design and analysis. So which means that in most of the cases, especially with the cell-based potency assays, is that there are quite a lot of variability there. So which means that a considerable amount of data may be necessary to get the statistical analysis right there. And also for the validation report that we will address the targeted validation parameters and their conformance to the acceptance criteria as well. So in respect to the current GMP guideline, that all laboratory records will have to be maintained and retained and ready to be to the inspector from the authorities as well. So the cell-based potency assay sounds pretty much straightforward and powerful tool to address the potency of our drugs. But there's still a lot of challenges there. For instance, that I just mentioned a couple of times in past few minutes, the variability is a big problem. So a lot of critical reagents are involved here. Since we are culturing the cells, they have FBS, human serum albumin, all the cytokines, growth factors we added in the cell cultures, and also the equipment that we are using, the multi-well plates, and also plate reader, flow cytometry, etc. All this will introduce the variability in our assays. And in addition to this, some of the advanced therapeutics, such as cell therapeutics or ex vivo gene-edited gene therapeutics, there are a lot of inherent variability as well with these starting materials. And in most of the cases, these therapeutics, we have a very limited materials for the testing as well. And also the stability, availability, and the behavior of appropriate reference control could be challenging sometimes. And the second biggest challenges we can find in this potency assay development is the expertise. Since we are talking about the complexity of this drug, mechanisms of actions, and the complexity of the assay format, so advanced scientific expertise and the time-consuming method development are required. And especially all the operators, analysts, including the reviewer, will have extensive training and with consistent performance as well. So most of the cell-based assays are labor-intensive. Since we started earlier, we will finish the experiments later, and which make the management of the technical resources much more challenging as well. So once again, that the challenges of the cell-based potency assay is the complexity of the drug system. So most of these advanced therapeutics, they have a complexity of these biotherapeutics modalities. Some of them contains multiple active ingredients, and some of them that the MOA may not be fully understood or characterized as well. And some of them also include multiple biological pathways. Here, just give you one example. For instance, for the gene therapies, We will test the effectiveness of the gene delivery into the cells. And also we will test this once the gene delivery into the cells, what the efficacy, the potency of the protein expressions, and also all the downstream effects of this transgene. So now I will leave the floor to Resh, that he will walk you through the potency assurance strategy right here.
So thank you. We've had quite a walk through some of the complexities that we have so far, and we're going to now spend a couple of minutes on the assurance strategy and give an example of how an assay will be taken through the stages, through late development and into the clinical stages and through into the commercial stage as well. The main point of this part of the presentation is to point out that this is actually very much a two-way partnership between the laboratory performing that testing, and I'm talking from the perspective of a CRO here. Cellbased does this testing to support commercial release stage and also clinical release for ATMPs. But the point is that the collaboration between those two laboratories is very much an enabling point for the success of these projects. It's really important that this two-way relationship works, and that the communication, the sharing of data, and the constant mutual review takes place. And it's incredibly important for that success. To move into the potency assurance strategy, the first point up here is that it's a science and a risk-based strategy, and we can take these science and risk-based strategies into three buckets. On the top bucket, we have a concept area. And our concept is also including the quality target product profile, a summary of potency-related characteristics, which also takes into account the clinical indication and route of administration, whether that administration be by subcutaneous, by oral, or by insufflation. All of these things come into account. The control strategy for the manufacturing process and product quality is a concept in itself, and the critical quality attributes that would be measured. Here I've just got the attribute. In fact, there are usually many attributes that are measured, some of which require a panel of assays through the drug substance, drug product stage for providing assurance around the product's efficacy. The critical process parameters that influence the CQA, such as the timing points of various downstream processing, are very important and come into that concept as well. So the control strategy is designed really to control the material specification. That includes manufacturing process parameters, hold times, run times, elution times, crystallization times, etc. And a panel of assays would be assembled to provide that in-process testing, so that each of the points of interest where the QP or the qualified person can sign off on each of these points will have a manufactured panel of assays to provide that degree of assurance in it. Each of those release tests provides access to the next step in downstream processing, further refinement of the drug substance or drug product to its final point. The lot release testing for licensed drug and drug product must be performed in potency release assays, which is a particular regulatory requirement. And each of these assays does require a degree of continued process verification, which usually takes the point of track and trend of the assay performance. And to the assays themselves, the suitability here is a major consideration. We've just heard from Yonghua that there are many technologies available, some of them new and exciting, some of them more, should we say, off the shelf and readily reducible, such as flow cytometry, performed by a number of different platforms. An assay should be suitable in that the technology used should not be limited only to one particular supplier. There are lots of lovely technologies out there that are proprietary, and where you have a proprietary technology that's not available from a different supplier, that would constitute a risk. If the company providing it goes out of business or suffers a significant restriction in a raw material or a process material that's available, that would manifest a significant risk to the ongoing availability of the assay and therefore have an impact on the availability of the drug. For instance, if you also had suitability of an assay consideration around the use of particular organoids. Organoids are a great technology, but to reduce those into a release assay would mean that you'd have to have a certain assurance around the supply of that, and that volume to be scalable to support the assay if it was used in commercial release testing. So the suitability around the assay would also come into the assay's resolution as well. If you had a certain resolution of an assay that would be tailored towards the critical quality attribute of potency or concentration, your assay should have a certain resolution of the assay in order to give you a particular point of reference around that to identify that quality attribute in terms of either picomolar, micromolar activity, etc. So all of that comes into suitability as part of the risk profile. For those assays, we need a reference material as well. Often at the early stages, this is a well-characterized lot of drug product. It's difficult to find for some bespoke products reference materials, so often the manufacturer will have a very well characterized material, and that should be a particular volume of that material to enable you to go through clinical trials and generate further batches of reference materials that can support through the commercial stage testing as well. Now, the qualification and validation of the assays is very important, and that can be done in a phase-specific manner, and I'll discuss that in the next slide. But there are particular points at which you need to have an assay that is taken from R&D into GMP arena, then taken into a formal qualification, which generally approaches the specificity and sensitivity of the assay, and then further into validation, sensitivity, specificity, robustness, reproducibility, and a few other factors that young Lara has alluded to in the early stage. They can be broken down into phase-specific activities, and that helps to reduce the cost, and all of these things that we're talking about do have an impact on the cost of goods, but it also enables a rapid progression of the assay and also evaluation of the assay in terms of its robustness and suitability to be taken into the phase 3 for support of the final phase and then into commercial. So qualification and validation is very important as a regulatory requirement here. We also need to think about changes and transfers of these assays as well. There are instances where we need to see revalidation, where we learn a lot more about the assay over time and also learn a lot more about the product over time and its production modalities as well, where we have changes in, say, late stage scale-up. We may need to finally tweak or manipulate the assays so that they're more in tune with the final product, which will be produced at a larger scale. That may well require revalidation. All of these new developments that we see require a formal risk assessment, see whether it would require revalidation or whether or not the initial validation is appropriate. And that would ensure the reproducibility of the final product itself and enable us to have the right analytics in place to ensure that what we're providing is of a suitable quality, is of a consistent quality, and therefore doesn't represent a risk to the downstream process or to the patient itself at the end point. As a point here, we've got an asterisk at the end here. As the point here for that lot release testing that we mentioned in the control strategy, at least one bioassay should be included. By bioassay, we really mean a cell-based assay. There are lots of instances where you may have a reductionist enzymatic assay that may be appropriate, but in this case, one bioassay should be included, and that's typically a cell-based assay. Now to move into the potency testing strategy, and what I've done here is to consider this as a progressive implementation of a particular cell-based potency assay. From left to right, we can read this like a sentence. We've got the new discovery, we've got the preclinical, we've got the phase 1, phase 2, phase 3, and then through into commercial stages. And at the new drug discovery, you've typically got drug candidate selection. The drug candidate will be selected, usually be selected on the basis of either an animal model or a structure or a discovery assay. And at that point, the discovery assay may be taken into further development into the GMP arena, where the assay will be taken into a qualification status. When that's ready, usually at preclinical or phase 1, the assay may be qualified. And to do that, we've had a formal assay development document, which is sometimes known as a feasibility document or a development document. That is typically out with the GMP process, but when that comes into the GMP process is by the formal qualification protocol. The qualification protocol should outline what's expected from the sensitivity and specificity aspects of the assay. It should also indicate how many qualification runs, and typically 3 or 4 runs would be appropriate, where there is a wealth of data from the development side. And then finally, a qualification report would be provided, which would provide suitable degree of assurance the assay behaves at that stage within an appropriate degree of sensitivity and specificity. I've used in here a graphic to show the continued process monitoring, because even after qualification, you may use those assays for several dozen more batches as we're going through phase 1 or phase 2. And that means that you must continue to look to see how that assay does perform, whether or not the sensitivity and specificity or any other indicators would indicate that there is a liability of the assay or you need to fine-tune or redevelop. Now, typically end of phase 1 into phase 2, there's often a tech transfer to the GMP site, for instance, a CRO that may be performing the longitudinal testing for end of phase 2 into phase 3 and through commercial. At that stage, it's typical to perform a formal validation. A formal validation will assess sensitivity, specificity, robustness, reproducibility, along with a few other factors as well. And that should be done at the site in a GMP environment where that drug will be tested. Okay, a validation protocol should be formally written. The validation runs performed, the validation report written and submitted as part of the CMC dossier. This should also be done before the initiation of phase 3. Now, where we do have some examples of drugs that may go through an accelerated program or an abbreviated development route because it's either an orphan drug or a novel drug or first in class, for instance, there may be a consideration around which time to do these qualification and validation aspects. And some companies or some manufacturers want to go directly to formal validation. That can be done, but there will be a particular bolus of data from the feasibility and from some monitoring of the assay performance before that to provide the right degree of assurance that a validation exercise will be largely a formality rather than a discovery period. Okay. All of that should happen before phase 3 typically. Phase 3 production of drugs should be supported by routine testing where that validated assay will be performed in a batch by batch by batch basis. Now, at this point in routine release testing, forward to the BLA, biological license application, it should also be performed on 2 sites in order to mitigate the risk of disruption of either supply of the service or particularly environmental disruption as well. We can all think of environments that may not be quite as conducive to routine testing, and some of them in developed parts of the world, where that risk must be formally mitigated. And typically that's mitigated by performing some aspects of the testing in Europe and performing some aspects of the testing in, for instance, the US or the Asia Pacific arena, to make sure that we can still provide that degree of assurance around the quality of the product on the different territories without having a risk to drug supply during the commercial stage. And that's also through the validated assay performing at GMP level. That should also be formally tracked and trended as well. At this point, it's important to know that any new banks of cells that are used in the assay don't have an impact on the performance of the assay, or any new raw materials used are well controlled so that the assay still performs within its specifications. All of that is a form of de-risking of that longitudinal commercial release testing and requires formal monitoring in that track and trend of the assay performance. So that takes us through a progressive implementation of the cell-based potency assay. But all of these things do happen with a degree of retrospective analysis as well. At some points we see for very bespoke products that there may be 2, 3, or even 4 assays used going through phase 1, phase 2, and phase 3. And it's not always apparent which of those assays will be the final one or the most appropriate one or the most tractable to use for them. So that strategy is often refined based on the quality and clinical data that we see. And in some cases, I've seen 4 assays taken through in phase 1 and phase 2, which would be reduced down to 2 assays or even 1 cell-based assay to support potency testing at phase 3. That requires a very, very good relationship and communication between the laboratory performing the testing and the manufacturer to provide the right level of insightful analysis on that data and provide the right choice. Often the experience of the CROs is very important and can be very useful for the manufacturer to make that final decision. And that's the modality that we use in Cellbased to make sure we have that communication and the mutual analysis of data so that it gives the best possible outcomes. And of course, a cost-effective outcome and a suitable outcome for the assay choice. That is part of that two-way relationship that is actually very enabling. It's not like a throw it over the wall, it's very much a detailed interaction between the two parties. Good. So at this point, I want to take the take-home messages, and they are that potency assays are required to ensure that quality of drug through the life cycle, and we need at least 1 bioassay. We minimize the use of animals where appropriate. The importance of the cell-based potency assays should be holistic rather than reductionist, should be versatile, but they should be speedy as well. The navigation of the development challenges means the right methods and expertise will be in place, not just in the development, but also where they'll be used for the longitudinal testing at commercial stage. And the lifecycle management of that data, including curation of data from the clinical stage and a commercial stage, is important and provides us with retrospective means to analyze that data and ensure that the assay is appropriate for its ongoing work. At this point, that wraps up what I wanted to share with you. I want to thank Yonghua for the collaborative nature of the preparation, and also my good colleagues at Cellbased for all the support, particularly the marketing team. And at this point, Ryan, I'll hand it back to yourself and we'll take some questions. Thank you.
Yes, thank you so much for this very insightful discussion today. I'd like to invite our audience to keep sending some questions or comments right now using the questions window for this Q&A portion of the webinar. And if you're attending via LinkedIn Live events, you can feel free to post your questions in the comments section. I've already received some questions from the audience, though, so we'll get started with those. The very first question I have for you here would just like to know, could you provide an example of a 3D culture for potency release testing?
Okay, so can you hear me well?
Yes, we can.
Yes. Okay, perfect. Yeah, so this is a very interesting questions. So most of this 3D culture platform has been recently developed for the drug candidate selection, for the screening, and also for instance, like a toxicity study using the human liver primary cells. And in our case, at Cellbased, that we have used 3D-like culture as well for the potency release testing. It's called the CFC potency testing. Also, it could be called CFU. It's a colony forming cell or colony forming unit assays. So those cells are cultured in the semi-solid media. Usually they are methyl cellulose or agar, and that contains growth factors, and we culture them for 10 to 14 days. And these assays have been widely used to evaluate the potency of hematopoietic stem and progenitor cells that could differentiate into mature blood cell colonies such as astrocytes, granulocytes, and macrophages, etc. So there are mainly 3 types of the drug products will be using this kind of a CFC or CFU potency testing. The first one is the stem cell therapies. And then the second one is the cord blood or bone marrow transplants. And then the third one is also the most commonly seen. That's the one they are ex vivo gene edited, the gene therapeutics. So all these three main drug products has been applied for this 3D culture media to look into the CFC potency assays. So those cells are allowed to grow in the 3D structures in this particular media. And that provide the critical quality attributes that Reg just discussed, and to provide the drug potency data to achieve the efficacies as comparisons with the data from the clinic. So this assays is very special because this not only quantitative analysis, it's also a qualitative analysis, since the colony number, size, and the type of the colonies are measured in this kind of assays. So that's the most common 3D potency assays that we have here for the batch release testing.
Great, thank you so much. Another question I've got here from someone wonders, how should the acceptance criteria for potency release assays be established?
That's a very important question, and it's a very good question as well, because assurance, so to make sure that acceptance criteria, that is to develop the scientifically sound and the regulatory compliant acceptance criteria. That's for our potency assays, to ensure the safety and efficacy of our drug product. So that means that the acceptance criteria should be designed to provide the confidence in the consistency of the drug quality across different batches. So the first factor we have to think about it is to understand the drug itself, and also the mechanisms of actions of this drug. So which means that the acceptance criteria should align with the mechanisms of actions of the drug product. And to understand how the drug product works at a molecular level is critical to determine what aspects of its potency should be measured. And for biologics, potency is often linked to biological functions. For instance, the antibodies, the binding to a receptor, eliciting a cellular responses such as cytotoxicity. Whereas for the small molecule inhibitors, it could be more focused on the chemical or enzyme activity as well. So the second factor to determine the acceptance criteria is that to collect as many as data as possible throughout preclinical and the clinical development. So early preclinical and the clinical studies can help to define the range of potency values that correlate to the efficacy and safety. So if a clear dose-response relationship is established in the clinical trials, then the acceptance criteria in the potency assay should ensure that the potency falls within the range that provide the therapeutic effects without causing toxicity. Also, that criteria can be set up during the assay development and validations. So during assay development, ensure that assay sensitivity, precision, and specificity are adapted to detect the meaningful differences in potency, and conduct a thorough assay validation. That's very important. And with all this data and during different stages with all these statistical tools like regression analysis, that can applied to the data analysis to ensure that we set up the acceptance criteria based on the realistic data from the clinic. And also the historical data that's important from the manufacturing CMC process, for instance, are important. And finally, that all these have to follow the regulatory guidelines from FDA, guidance for industry on potency assays, as well as the ICH, International Council for Harmonization's ICH Q6B guidelines on specifications to set up the acceptance criteria are important. And not all of this cannot be done without the statistical analysis. Has to have the thorough statistical analysis strategy in this case. So thank you again for this question.
Oh, wow. Thank you so much for that very detailed answer. I'm sure they appreciate it. And thank you both for this wonderful presentation today. Of course, we have reached the end of our time. If we cannot attend to your questions, know that the team at Cellbased will follow up with you. Or if you have further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link in the chat box with a link to view the recording of today's event, which you can share with your colleagues so they may register for the recording here as well. And for those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event to gain access to features such as viewing the recording and more. Now, please join me in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Bye.
Thanks. Bye.